how phytotherapies affect the overall health of hyperglycemic induced C. elegans. This research is important because statistics showed a very heavy use of herbal medicines amongst diabetic and infertile patients. From this, we wanted to see how various phytotherapies would affect the overall health of hyperglycemic induced C. elegans, and we hypothesized that a concentration of 0.01 grams per milliliter of ginkgo biloba would have the biggest effect in bettering the overall health. This would be shown in an increased egg and thrash count. So essentially how we did this project was we induced our phytotherapies and glucose into LB broth, and we used 3 steroid dilutions. We then autoclaved, added the E. coli, seeded the plates, chunked the C. elegans onto the plates, and then we used a stereoscope and a phone to take 15-second videos of the C. elegans and used a clicker to calculate the tail thrashes. Then for egg count, we used a sterile worm picker to pick L4 C. elegans off the plates and added to 10 microliter drop of 5% bleach and used a stereoscope to count the eggs. For our control group, we used the same methodology just without glucose in step 1, making them non-hyperglycemic induced. So our result entailed that a concentration of 0.01 grams per milliliter of ginkgo biloba had the biggest impact on increasing the thrash count in both hyperglycemic induced C. elegans and wild-type C. elegans. When comparing both graph data values, there was no statistical significance, meaning the spices affected both the hyperglycemic and wild-type C. elegans, due to the hyperglycemics having a higher standard of the mean shown here. The hyperglycemics were seen to be affected more by the spices, bettering their overall health. So our results for a concentration of 0.001 affecting the egg count entail that turmeric had the biggest effect in increasing the fertility of these hyperglycemic induced C. elegans. Although there was no statistical significance, we're still in the process of researching egg count, so comparing these values to a control group would give us a better understanding of what's really happening here, and we expect as we input the other concentration there to be a likely statistical significance. So in conclusion, our hypothesis was invalidated in terms of ginkgo biloba increasing the egg count, and our research can be implemented as potential aid in bettering the health of fertility of diabetic individuals using phytotherapies, and these are our references.